Good evening, everybody. Thank you so much for coming to the CNR in March. So I just want to welcome our speaker, Anna Bonkhoff. So Anna is, I've only met Anna once or twice, but I've known about her name because she worked with Christian Grefkes in Cologne in an area that I did quite a lot of work in many years ago in brain imaging in people with stroke, both structural and functional brain imaging. So Anna's name definitely came to the fore in that field, so I knew who she was. Currently she's working at Massachusetts General in Boston, doing a neurology fellowship, which sounds very arduous. Over my own experience of knowing what it's like to be a fellow in the American system is watching Grey's Anatomy. But I presume it's something I used to watch Grey's Anatomy, I haven't watched it. But that sounds hard work. But she's continuing to do work in brain imaging and stroke, and so she was over in Europe, so I nabbed the opportunity to get her to come to London and give us a talk about anything she wanted to talk about. So now she's going to talk to us about novel ways of predicting outcome after stroke, which will hopefully involve a lively discussion afterwards about whether we want to do prediction after stroke, because my experience of talking to therapists, of which there are some in the room, they don't actually want to do prediction at all, because what if it's wrong? Anyway, we'll talk about that later. Dan, thank you.
Anyways, I'm delighted to be here. I did a very brief internship at Queen Square in 2017, so it's a while ago, but I'm always very happy to be back here, and also now excited to present a little bit of my work with different collaborators, one of them being here. So Howard and I worked on many of the projects that I'm about to present. And I'm happy for you to interrupt me for questions, or we can do them at the end. And so I just have to find out how to advance the slides. Okay. So the first part of the talk will be about the prediction of upper limb impairments post-stroke. And then later on, I'll talk a little bit more about imaging work and my most recent project. And so if we get to this, I'll try to stay on time. So after all, one of the first projects that I started working on was this proportional recovery project. And so I think probably most of you are familiar with the proportional recovery rule that was introduced quite a few years ago by now, and is beautifully straightforward, as in, so recovery after stroke can be predicted or computed by taking 70% of the recovery potential, with the recovery being the change between the initial and the final score, and then the potential being the maximum that you can get to in your initial impairment. And so after this rule, let's call it, that was introduced, I don't think I know exactly which advanced. It was actually found to be the case not only for upper limb impairment, but also impairments of the lower limb, and then other domains that could be affected, so attention, language, cognitive domains. And it was quite convincing in all the individual studies. But then some teams, so actually two at the same time, and one of them being Howard Bowman and Tom Hope as the driving authors, realized maybe there are some statistical problems relating to this rule. And so I'll touch upon that briefly just to have it mentioned, but I won't go into the math all too much. So the two effects are called mathematical coupling and ceiling effects. And essentially what those two effects lead to is that you will always derive that this recovery rule is true, whether it is actually true or just a a statistical fallacy, essentially. And so I kind of came in at this stage because I was wondering, okay, if we know this, how we previously derived the recovery rule doesn't really tell us if it's true or not, how can we find different ways to say something about what's the pattern of recovery? And so this is just to briefly illustrate what's mathematical coupling. So basically the problem arises if your final score doesn't have as much variability as your initial score, which for many of the scores we obtain to measure impairment or function is the case because there is a maximum score you can get to, and at some point we don't have the resolution of saying is this patient still improving or not, because he or she might already be at the maximum function. And so the ceiling effect in mathematical coupling is like automatically given for most of our scores. And a lot of different people published on these fallacies after all. And what we then came up with in terms of how do we investigate it further was to get a pretty decently sized data set in contrast to the original studies. I mean, it's always very difficult, and there's a lot of energy going into acquiring these data sets, so getting a bigger data set is quite complicated. And it was, though, our goal for this project, and we then kind of invented different scenarios or ran simulation studies to see what can we do to address these statistical problems and resolve them. And we then eventually wanted to be able to say, what's the pattern of recovery? Is it proportional to the potential as initially thought or not? And so walking you a little bit through this. So we were able to get a data set with 385 patients, which, as the figure shows, was made up of 6 individual studies. So we were fortunate to have several research teams either share their primary data, or there was a team in Canada that had actually visually extracted this recovery data from plots in previous studies, so it was kind of secondary data. And what you're seeing is really just the initial Fugl-Meyer score, which is plotted on the x-axis, and then the final Fugl-Meyer score on the y-axis, with them being obtained in the first 2 weeks after stroke and then 3 to 6 months after. And I don't know if can you see my mouse? Yeah. So I think the problem with modeling recovery with this data is really this clustering here, because all these patients basically improve to the maximum already. So it's difficult to know would they have improved further if our score allowed like a different resolution at the upper end or not. And what we then learned from simulation studies is that while most studies were focusing on this entire range from Fugl-Meyer initial scores of 10 to 66, you kind of have to exclude actually the patients that were only presenting with mild deficits in order to then fit your models. So it's basically, unfortunately, saying that your model is only can only be accurate from 10 to, I think we had 45. That was the result from our simulation study. So we had artificial data that could then give us some clues on what's the maximum score we can model. And this non-fitter-fitter split basically comes from the literature and how the models were derived previously, because very severely affected patients were not included from the very beginning. And so having the data set to work with, like knowing that we only work with patients in this Fugl-Meyer initial score, we then had different hypotheses on what the recovery could look like, with that being it could either be proportional to the lost function or proportional to the spared function, which basically just means you measure it in different ways, like either you have the maximum score and you subtract your initial score or you take your initial score. So those are two proportional patterns. And then it could also be the case that all patients just improve by the same amount. So that's the constant recovery part illustrated here. And so this looks very complex, but I think what's just important to take away is that after addressing the statistical fallacies of the initial models, we still derive that the proportional to loss model, so the proportional recovery pattern that was assumed previously, is still the pattern that we find. So it kind of recovered that. The change that we saw, though, was that after all, we couldn't explain recovery very well. So initial studies had explained variances of up to 90%, which, I mean, would be amazing if you could explain stroke outcome to that extent. And what we found with our data was, roughly 30%. So it does say actually just one single value, so just the Fugl-Meyer initial score, does give you an idea of what the recovery will be, but it's just not very high. Or I think it would be difficult to clinically work with this prediction. And so after having looked into this, I was kind of always a little bit unhappy about throwing away all the patients below a Fugl-Meyer score of 10. And so in our next project, we looked into, can we actually model both the very severely affected patients and the moderately affected patients at the same time, and is there a benefit to it? And so in our subsequent project, we did not throw away all the patients with lower initial scores, but we did include them. And what we interestingly, that just illustrates it, so that we went from not including the non-fittest to then in the middle to the right side where we included everyone. And what we found is testing, should we model them together or not? It was actually more, we could increase the explained variance, so explain the outcome better if we modeled both groups at the same time. So that was actually quite reassuring. This just illustrates how the data that we used, it was basically all the 6 data sets we had previously, and then we had 2 different teams joining us at this point. So on the one hand, it was data from the FLAME study and then data from Boston, where I then already was at this point in time. And interestingly, so if you contrast modeling everyone as one group versus what happens if we model it as 2 separate groups, it was our models favored the situations where you do have different recovery patterns for the very severely impaired versus the moderately impaired. And so, which is here illustrated by basically the lines kind of speak for what kind of recovery patterns you find in the ones in dark blue are the ones that are severely affected, initially in the light blue ones, the ones that are moderately affected. And so the moderately affected ones always recover according to the common proportional recovery to loss function. But then the ones that were severely impaired recovered proportional to their spared function. So actually the patients that had more preserved function recovered a little bit better. So it was kind of the other way around for the different groups. And having run these studies, we were then wondering about whether intervention does change anything about the recovery patterns that we find. And since we did have the FLAME study data, and I feel like you are probably all aware of the FLAME study, so it's a study from now a few years ago. It was published in 2011 that did show that fluoxetine increased or enhanced motor recovery when measured by the Fugl-Meyer score. And I know that there's a lot of controversy about it by now, since there were subsequent studies that couldn't really corroborate these findings, even though they had different outcome measures. But at least for this data, we kind of have the initial studies showing that there is a significant difference between the intervention and non-intervention group. And so what we did was to now include both the intervention and non-intervention study in our model. And so just to say, so the model that we used was a Bayesian hierarchical model, and one of the very convenient qualities of that model is that you can have these subgroups and you can all model them at once, which increases your variance. So since you have more data to model outcome with, it overall predicts the outcome better. So yeah, it's kind of models I really liked to use for that data. And so this is unpublished data, I think. So what we found was that the fluoxetine arm, or the patients that received fluoxetine, did have an increase in how much proportional recovery they had. So they did recover proportionally more than the ones that didn't have the intervention, which was, so I mean, I think at this point, I am always wondering about what does this tell us about the brain? How is this actually, what are the processes? So I unfortunately can't say anything about that because all we looked into was the change in Fugl-Meyer scores. But it kind of hints at that something different happens in the brain to allow this recovery to happen. And the next step, so having gone through these different iterations of acute patients, was to now look into, okay, can we find similar recovery patterns also for patients beyond the acute phase of stroke? So everything I presented so far was stroke patients that were included in studies within the first 2 weeks of stroke. And so for this next part, we looked at stroke patients that were 6 months and beyond their stroke. And so on the one hand, this is all also still unpublished data. And I guess Howard is aware of all these studies, and I wished to have already written it up, but residency kind of interfered, and so things take longer than I wanted. But so the data that we worked with is from the VNS rehab study that was also partially run in Boston, so my collaborators there were able to share the data. And just to give a quick overview, so it was basically patients, as mentioned, 6 months after stroke, and they had to be within a certain range of Fugl-Meyer score, so I think it was 20 to 50 at this point. And they then received treatment, I think, over a 3-week course or 6-week course. And so what I analyzed was the data obtained right before the VNS stimulation and then 3 months later. And that was at different points in time for the patients post-stroke, but all in the chronic phase. And what they had already shown in the VNS rehab study was that the stimulation group did significantly improve with the treatment. And this just plots the initial and end scores for the 123 patients that I worked with. And what we found was that all these, and I'm sorry, it's not the most beautiful visualization, but what we found was essentially that all patients recovered in a constant pattern. So everyone, even the ones in the the stimulation group, gained just a constant amount of, or just a certain number of points on the Fugl-Meyer score. So it is very different to what we see in the acute phase where it is proportional. And then the VNS treatment boosted this constant score. And once again, I guess this reminds me of okay, the processes in the brain somehow seem different, but it's not something I can prove since I've not looked into the actual brain data. It's just projects that are going on at the moment. This just illustrates the numbers. And then the second arm of the study was focused on data from the NICHE trial, which is a TMS study that actually did not show any significant gain in or the Fugl-Meyer function or score. So effectively, what we found for this study was that our model also suggested to model everyone, even in the treatment group, as one. And they all still had some constant recovery, but given that there was no gain for the treatment part, we also didn't find different pattern or numbers for constant recovery. Okay, and so this kind of concludes this first more focused on upper limb impairment part of my presentation, with the conclusions being that we find different recovery patterns for different subgroups, depending on, is it acute, is it chronic, is it in the moderate range or severe range of impairment. And so for the acute groups, it's always proportional. For the chronic ones, it was constant. And yeah, so to give you like a brief primer on other work that I was fortunate to work on in Boston, and before Boston in Cologne with Grefkes and Grefkes, I'll dive into prediction of stroke outcomes now, more based on imaging markers. So it's very different in the way that we use the imaging data, and unfortunately don't have as granular outcome markers anymore. So most of the outcomes I work with were stroke severity or a modified ranking scale. So I've yet to merge both of these approaches, basically. And so just talking a little bit about like what, and I guess that's probably repetitive as it's a common theme. So what do we want in terms of precision medicine? And I think, so the goal, of course, is to provide an individualized prediction for every patient, which for stroke seems quite important, as we do have so many stroke patients, and there is a lot of burden of disease after a stroke. I kind of went into the imaging field as at the time that I was starting my research, there were more and more growing data sets that, so I knew I wanted to do something with machine learning, and then, you know, okay, you kind of have to work with bigger data sets. And so we have big data sets in stroke a little bit, but they are still small in comparison if you look into the actual machine learning literature. So data sets that are available currently are, for example, there's one local one here in London, even that at the time that I worked with had 1,300 subjects with just structural scans, basically. And then there are groups in South Korea who are accumulating a lot of scans. I think they are now at 6,000, so the numbers are not quite accurate anymore. And so I worked mostly with data that was gathered in, or more agglomerated in Boston. So it was MRI Genie with 3,000. And so there was some opportunity to now work with bigger data sets. But even having these data sets, the problem you encounter is that imaging data is so high dimensional that it's the initial problem you have to solve is how to break down this high dimensionality of your data to even then generate any predictions. And this is kind of what I focused on in early project, and where I took a lot of different people's advice of how to go from 200,000 voxels in the brain to fewer dimensions. And I'll highlight one project here that I did with Parashkev Nachev, who some of you may know here at UCL. And our goal was really to use this UCL data set of 1,333 stroke patients and investigate, are there common patterns? And if you ask stroke clinicians, who are generally look at lesion patterns, there are clusters of what strokes can look like, because we only have so many vessels in the brain and they are occluded. So all of this is ischemic stroke data. So your strokes do kind of look a certain way. But the idea for the project was to not have this hypothesis at first, but have it be proven by the data itself. And what we did was to use all these 1,300 stroke lesions and use just mere lesion location. So it's binary data. You have a 0 everywhere in the brain where there's no lesion, and the 1 where there's a lesion. And we use different dimensionality reduction algorithms And the one that worked best for us was non-negative matrix factorization, which is very similar to just principal component analysis. So it's just one specific algorithm. That's, though, very suitable for imaging data as it's just positive. It's 0 or 1. And then we used a machine learning algorithm called TISNI. And yeah, without now going in further detail, what we came up with was that this is what our 1,300 strokes looked like after using all these fancy algorithms. And we kind of clustered them, so I had individual groups. And I mean, it did look pretty, but after all, it was difficult to just know the value of it. I mean, it is a lower dimensional derivation representation. So if you remember, initially we had 200,000 voxels, and now we have 2 dimensions because it's just like the x and y axis that differentiates where in the space is your stroke patient. And if not the 2 dimensions of x and y, we had, I think, 21 groups, so that's the different colors. And actually, what then ended up to be the case is that these different algorithms sorted the data in a way that made sense. So if we average the lesions of all the patients in the different clusters, we just obtained very, all the different stroke lesions that you would also think about. So we found the ACA strokes, and then all kinds of different MCA strokes and subcortical strokes, posterior strokes. So the important part here is that we derived this automatically. So it wasn't that we knew, oh, this is what we want to end up with, and these are the lesion types that they need to be, but it was really just like putting in the data and using the different algorithms and then coming to this distribution. And the then important part is, can this representation be useful for the prediction of stroke outcomes? And we kind of started looking into this in simulation studies, and all this is saying is that so if you use our lower dimensional representation, you can boost your prediction. So this was more of, we didn't have outcomes for this data set, real outcomes, so it simulated outcomes. And what we found was that using this lower dimensional prediction does give you a gain in how well you can predict the outcome. Though I must say it's not as powerful as actually using an even higher dimensional representation. So let's say if you just took the results from the first step, the non-negative matrix factorization, and used 50 dimensions, you could predict it even better. But there's always a trade-off in terms of how well do you want to interpret your representation and how well do you want to predict it. And so the representation that you can see on the left is kind of the sweet spot in terms of like, we know what we are seeing. We can say this is this kind of stroke, and then it still helps us with prediction. So yeah, this about that. And then I spent a few last minutes on a topic that I've looked into in the last few years, and I hope that I'll be able to look into it further. There's some controversy on what research will be funded going forward in the US. And I probably shouldn't be saying this aloud, but I think, so what I've realized for the data that I've worked with is that it did actually make a difference in how we model stroke outcomes. And this is just a general overview that I've created with some of my research friends this past fall, that there are reasons you could think that it does make a difference because they are different sex hormones and sex chromosomes, and they do have a whole lot of effects on how the brain functions, which is mostly a word maybe when it's a healthy brain, but becomes more obvious once you enter the space of diseases. And there are a lot of unique studies, more so also in rodents and non-human primates, that show the effects of hormones. And I try to show you on a few slides it did make a difference for our prediction. So one of the first projects I looked into, and this is now with MGH, or with data that we had available in Boston, was that if you use lesion location, so basically like in the previous project that I showed you, and then try to predict stroke severity, so just the NIHSS, acutely after stroke, is there a difference on how different stroke patterns predict the stroke severity? And what we found was that this particular lesion kind, so that's mostly lesions affecting the posterior part of the brain. So if these areas are affected and only on the left side of the brain, then strokes tended to be more severe only in female patients. And this is something that so I found for two independent data sets in Boston, and which was since also replicated in a data set with 6,000 stroke patients from South Korea, at which point I do slowly believe that there's something about it. I'm not saying this explains everything about why outcomes are different between men and women, but there seems to be something specific about these areas. And unfortunately, my data can't really tell us what it is. But what I've since been trying with help from, so this previous project was supervised by Natalia Rost, who was my mentor in the last few years in Boston. And then this next project is with Michael Fox, who is also in Boston, uses lesion network mapping. And so the idea of lesion network mapping is very neat in the sense that you only need information on the lesion location from your actual patients, and then you merge it with functional connectivity data from healthy participants. And what it allows you to say is that you know the location of your lesion, but you also know how connected it is to the rest of the brain. And so we used this approach of lesion network mapping to then look into what can explain the differences that we see. And what we saw was that for this particular posterior lesion pattern that was associated with more severe strokes in women, it was also the case that only based on the functional connectivity from female participants, we had like more global functional disconnectivity. So it, for some reason, led to more disturbance in the entire brain than for male patients, which is also only another hint of what's actually been going on. But so I've then been very fascinated by the lesion network mapping approach, and this is the last project that I'll highlight, which is also not yet published, but what we then thought about is, okay, can we use lesion network mapping to explain more differences between men and women with respect to symptoms after stroke? And so one phenomenon that's very sex-specific in frequency after stroke is post-stroke depression, with more women being affected, both if it's just major depression or after stroke. And so there's that kind of hints at like that there is some mechanism that puts one more at risk than the other. And so this was, so Michael Fox lab has already used lesion network mapping to explain post-stroke depression in general. And what they found was that based on their data, there's one spot in the left frontal lobe that if that's functionally connected to your lesion, you're more likely to experience symptoms of depression. And this is just like explaining our project approach. And then what we found was that actually if you just compare the lesion connectivity for male and female patients, then it's really affecting the entire brain, with female connectivity being both more positively disturbed and more in the negative direction. And this is just something that wasn't really looked at before, because usually if you do lesion network mapping, you just use the entire data set of male and female patients or healthy subjects. And overall, this just illustrates what we saw on the previous slide, is the actually significant areas, which was the same for 3 different data sets, so we kind of tried to replicate it right away. But what we then found was that the lesion connectivity that we derived was more was more strongly correlated to fingerprints or previously established circuits of depression than for male patients. So overall suggesting that there's something about the connectivity in the female brain that, is reminiscent of the patterns you find for depression. And in the very last step, we then looked at like other different patterns. And so this is only exploratory because my data set was very small, but what we found was basically that the hotspots where you should stimulate in order to treat depression post-stroke is slightly variable. So it was a little bit more medial for female stroke patients, as illustrated up here. And it was bilateral, while it was only left lateralized and more lateral for male patients. So just suggesting that if we use like individualized stimulation areas and TMS treatment of post-stroke depression is often done in the US, it could maybe boost the outcome. And I think that's as far as my talk goes. And I have a lot of people to thank. So all this work was always in bigger groups of people, amongst others, Howard Bowman and Tom Hopier for the upper limb recovery work, Prashkev Nachiv, I've mentioned that one project, and then more who allowed us access to their data and gave recommendation on analyses and the strategies thereof. And then the two labs that I'm part of, so Michael Fox's lab that's at the Brigham Hospital and Natalia Rost lab at MGH. And so I hope that wasn't too fast or too much.
No, it wasn't too fast.
All right.
So questions, come on, you know the deal. No questions, no wine. David.
Thanks a lot for this amazing talk. One question about the...
Thanks. So a question about the recovery prediction from the acute phase. You showed these two different patterns with the more severely impaired or non-fitters and then the like mildly or moderately impaired patients. And I was just wondering, what do you think about the value of adding biomarkers to this modeling? I mean, it's known that, for example, MEP positive, negative has a great predictive value for the severely impaired group. There are other imaging biomarkers. So what do you think, how much could this prediction improve with additional biomarkers?
Yeah, so I guess I can only offer you my best guess because I haven't worked with the actual data. I think, just using one single behavioral score can probably be boosted by using more markers. And I feel like whether this is lesion location or maybe even lesion size, and then TMS or like stimulation data will all enhance it further. And I hope that's something we'll look into. I think what our calculation showed is that we can only predict such a small amount of explained variance that we'll hopefully be able to improve it. The high dimensional case. I'm still undecided about what my expectation is, how well we'll be able to predict stroke, because I'm not sure if everything that we know how to capture at the moment will be enough, or whether we have to be more creative about maybe merging different domains and not only having the motor outcome, but also adding your attention and just becoming really broad will allow us to get there.
You would agree that probably different groups would emerge because it looked like very uniform, the very uniform patterns for the different groups. But if more markers would be added, then different groups could emerge from that.
Mm-hmm. So I guess the only thing I can say based on an analysis I've done is to look into whether we can split up these two groups into even more groups. And these were just exploratory, so we haven't described them in depth, but we always came down to these two groups. They seem to capture most of the variance. So I feel like there are different mechanisms at play, and the recovery happens differently. But I'm sure we'll also be able to define even more subgroups, especially for the severely impaired.
Thank you.
I guess with the prediction, I guess the issue is simplicity as well. The more markers that we add into making predictions, the harder it's going to be to actually implement it in clinical practice. I guess the ideal world is where somebody comes into the acute stroke centre, the pattern of damage is delineated, and then that is enough to make some useful predictions that kind of help something about to do with clinical reasoning and profiling of those patients. If one then has to kind of wheel TMS machines in or wait for blood tests, then maybe it makes it too complex. So it's always a payoff, I think, between the simplicity and the how easy it is to do it. Yeah.
Thank you. Great talk. I thought it was very interesting to see the constant increase in the chronic patients, so that there we don't have anything proportional, but we just see that an intervention basically amplifies that constant gain. Mm-hmm. And you already hinted at what could be the brain mechanisms. What are your thoughts?
To be honest, I don't know. I wish I could look into it. We don't. I think for the VNS rehab study, we do have brain data, so somebody, I'm not sure if I will be the one or somebody will be able to look into what the different mechanisms are, but I can't really offer you a concrete scenario here because it's not something I've personally looked into much in terms of brain correlates.
No, because I
think it's an exciting scenario. So I think one of the future work I'm hoping to do is just find the mechanisms for actually recovery generally. So I think it's one thing to model the changes in motor function, but I think it could also be the case that you have different maybe brain networks, let's call it that for simplicity, that support your recovery overall, like in all kinds of different domains versus only motor. So that would be very interesting to look into, but it's not something I can say based on data.
Thanks.
So I think one of the interesting things from the proportional recovery stuff is, and I don't know, you may have done this analysis, but I don't think you showed it to us, and it might tap into Seb's question, is, I mean, the kind of the 70% thing is controversial. So it's interesting that you showed that it is there, but actually it's only accounting for 30% of the variance, which is nice. But the most striking result that ever came out of the proportional recovery stuff to me was those severe patients, and that they basically at some so initial severity, and this is in upper limb and it's all in the Fugelmeyer, although it does cross domains to a certain extent, is absolutely no use at all in predicting future recovery. Okay, so if you're moderately affected, we kind of know how you're going to do. If you're mildly affected, we kind of know how you're going to do, which fits with clinical practice. But if you're bad at the beginning, some people do really well and some people do really badly. So did you ever look at the difference in any kind of imaging parameters for the severe patients who kind of split off into the good recoverers or the bad recoverers? Because I think it speaks to something that's really, really important in this field, and that's the difference between the factors that are important for initial severity and the factors that are important for recovery. And those are two different things. And the fact that you can start off really bad and you can have many different cohorts, but you can have good recoverers and bad recoverers tells us that. So it's almost like the pattern of brain damage. The structure maybe sets the scene for how bad you are at the beginning, but for want of a better word, plasticity, or the mechanisms that underlie so-called spontaneous biological recovery, are the thing that then determine what your trajectory is going to be. I know that wasn't the question, but is that?
No, I mean,
there was a question in there.
It will be, or I think there's some work going on also for the severely impaired, and there are some really promising markers. For example, if the corticospinal tract is still intact, I think you have, on average, a better recovery than if not. and I mean, it's something I, at the moment, personally only observe like on the side of treating acute stroke patients, which I still find fascinating because we have some that have severe impairment initially and then recover within a few days. And you're just like, oh, what happened? And in others, it's not, but it's not something I've investigated myself.
Thanks very much for that. That was fab. I just wonder, I know at the beginning of your talk, you talked about the FLAME study, and then at the end, you came back to some of that post-stroke depression and connectivity mapping, particularly more in women than men. But is there any way you can pull those together? I mean, is that something you guys will then look at the next stages? And is that something we missed in the original bit that you're now looking at?
I think that made me even more excited working on the post-stroke depression data, and I think they are really fascinating studies actually that are decades ago that showed the different pathways for serotonin or it made sense in that way that there's more plasticity or recovery, but I've not actually linked these two projects. I think there's something about it, and it's probably so I feel like it's all these processes are so complex, and serotonin and then estrogen and all the other hormones, they also interact, and their receptors interact or work better or worse in the co-presence of something. So I think there's a lot more to explore, also mechanistically. That will be very exciting. But yeah, I can't, unfortunately, answer you the question really well otherwise.
Hi, thanks for this great talk. I actually have two questions. One is, again, related to the proportional recovery and linking it back to the end of your talk. Are there sex differences in these trajectories?
So, I mean, I couldn't investigate it in our data since we didn't have information on the biological sex. I don't think there is much described in terms of sex differences for motor recovery. I don't know, Nick, if you have a different impression. But so I think that motor recovery seems to be the same, which also is the same for language. For example, I don't think that for these domains that there are any larger differences described clinically, which is also why we went for depression post-stroke, because we knew that there's a clinical difference, and so it's more likely that something different also happens in the brain. I think cognitive deficits could overall be interesting since dementia does have a sex predilection also, but not for motor as far as I'm aware.
And about the last results you showed, which are fascinating. So if I understood correctly, there is basically a difference in the networks that are affected, which are sex specific.
So at least what our data suggests, and I guess that's still within the limitations of running exploratory studies only, we did find different hotspots, which I suppose is the most important part if you then want to invent a treatment or stimulate, for example. So I'm hoping that we can repeat this analysis with bigger data sets, and then
But do you have an idea why that is? Is that are there known sex differences in some anatomical features in terms of, say, the connectivity or the vasculature? I don't know, like magic.
Yeah, so I think like if you look at anatomy of the human brain and also connectivity or everything in the human brain, you find very, very few really substantial differences. So everything other than brain size is actually pretty similar. So brain size is the only difference where I think, men do have 8% bigger brains than women, and that does change a little bit about the ratio between between gray and white matter. But other than that, even if you look into functions of the human brain, there's very little huge difference that you find in meta-analyses. And so for me, and especially, so we wrote this one review, and going through the literature is that I think a lot of differences arise once disease starts and progresses. So I think the mechanisms do differ a bit, and I think that's particularly interesting also for Alzheimer's dementia, where there's definitely a link to menopause and then hormonal status, and there's a lot that needs to be explored still. But I think this kind of then tells us a little bit about that probably the molecular processes are a little bit different and how the different hormones interact, which we probably don't capture with just imaging, brain imaging.
Anna, thank you so much. That was really interesting. I have to admit, a lot of it went really over my head, so forgive me if this question isn't. But going back, I think, a little bit to what you were saying, Nick, with those really very impaired gang and the kind of fact that you get these people doing really well, and is there not a lot to do with neglect? Do you see that within the kind of connectivity? Is it those kind of, those patients who develop?
That's actually a really, interesting question that hopefully, so it's not something I've looked into or know that the connection was drawn, and I think that's a very exciting space for future studies, and hopefully there are coming more and more that actually draw those connections. So I know that one of my collaborators in Boston, for example, evaluated just, I think, motor improvement, function improvement, linked to cognition or attention, and it makes a huge difference. And so I think that's something that probably we should always incorporate in our models, but sometimes you just only have one information but not the other. So I think pragmatically it's often difficult to tell, but I think there's a huge influence.